Assalamu alaikum, everyone. Happy Friday and good afternoon. My name is Raffi AlMutawa. Many of us have met before, and there are many new faces. So, hello, everyone. I hope preparing for the finals going well and that you're all studying and working hard. So, today's topic, by popular demand, is cell communication. And if you were here in pre-med, you would probably recognize some of these slides. Spoiler alert, the jokes are probably still the same. So, my contact details are here in case you have any other questions. And of course, please go through my notes because they'll have every explanation that I guess is on here. So let me just put this away. All right. So this is our objectives for today. Now my assumption is that this objective, the introduction to communication modes, I think this one's quite easy. I think everyone has a decent idea of it if you've been through this lecture before. We're still going to go through it regardless. The principles of cell signaling, it's also quite an easy objective. We'll go through it regardless. Now, I think here, the molecular switches in speed. Oh, let me turn on my laser pointer. So, there it is. Yeah, so the molecular switches in speed, I feel like this may give some people a little difficulty. It is a very important objective. And I think my assumption is that GPCRs, cyclic AMP, and inositol phospholipid, I think that's where everyone's having some difficulty. But we're going to go through everything regardless, and we're going to go through it until everyone has a good understanding. So just to start off, what is a signal? And for you physics nerds, I don't mean this in the electric way. So you can write it down in chat, whatever you think. I'm going to try and interact. I know it's difficult, but I just want you guys to tell me what a signal is in your own words. Message, stimulus, message or stimulus. Okay. I like the neutrality here. All right, a way to transmit, a form of communication. Okay. So for some reason, this isn't moving away. Apologize, but okay, that's worked. Yeah, so a signal is essentially a message. It is a stimulus. It's everything you guys have said. But just to keep it so simple, a signal is anything that tells anything to do anything, all right? For example, breathe manually, all right? Now, the signal is telling you to breathe manually, and then you started breathing manually, or even if you didn't respond to that, it's still a signal, all right? And let's look at a few examples. So this signal means you're turning right. This signal means you've got Wi-Fi connection. And this signal means we're taking over Poland, although for some reason they stopped using this in 1945. Not quite sure why, all right? But let's go through some basic definitions. So in our context, a signal is anything that tells cells what to do, because our topic is cell communication. And if our cells don't communicate, well, we're gonna die. We also have these signals, they're transmitted through a form of a molecule called a ligand, all right, or a ligand, however you want to pronounce it. Now these ligands will bind to something called a receptor. The receptor just binds the ligand. And then we have this thing called a secondary messenger. This just responds to an outside signal. And then we have something called amplification, which is basically increasing the size. Now, just for a better idea, just for some better context, let's talk about our cells. So I don't care what you think this cell is called. I don't think this cell is important, but essentially what I care is this process right here. So our cell, this cell right here, wants to signal another cell, all right? To do so, it uses a molecule called a ligand or a ligand. So it sends these ligands, which will bind to the receptor, and that triggers a response, the signal. All right, simply enough. And there are many ways which our cells do this. So it could be through these modes, all right? You've probably seen this picture before. So this is two cells touching each other. They're directly fused to each other. So this we would call juxtacrine or cell-to-cell communication. Right here, we have a cell communicating. It sends this ligand right here, and then this receptor is on its cell. And then we also have another ligand here, which is sending a signal to another cell right here, which is very close. So because it's signaling to itself, this is called autocrine. And because it's signaling to its neighbor or a local cell, this is paracrine. You'll come to see that these two methods are also considered short distance communication methods. And we'll talk about that in more detail. This right here, we have a neuron communing with a cell. So this would be nervous or neuronal or neurocrine. whatever makes you happy. And then here we have an endocrine cell. And I believe you guys have a lecture about glandular epithelium, so I'm going to save that for Ala. Or you have a pal on glandular epithelium, I'll save that for Ala. But basically with endocrine communication, you have a ligand goes through the bloodstream and reaches the target cell. So this is called endocrine communication. And you'll see that these two methods are actually long distance, right? Let's think of another way to think about this in case you're still lost. So I like to think of this as, actually we'll talk about details and then talk about the analogy of basketball. Let's start off with juxtacrine communication. Again, we said that this is physical direct contact. So we have these cells which are directly connected to each other, but you'll see it's not between the membranes. The membranes actually have a gap between them. Really, what connects them are these junctions, and we call them gap junctions. There's a gap right here and a junction right here. So it's a gap junction. Now, the way I like to think of this is called kissy-kissy, huggy-huggy communication, dependent communication. And the reason, and I didn't come up with this name, by the way, I believe it was Dr. Ann, but the reason we call it kissy-kissy, huggy-huggy is because they're directly contacting each other. And that's what you do in a kissy-kissy, huggy-huggy situation. Now, dependent, why? Now, I said that communication is the basis of life. We only live because of communication. Our life is just a series of cells communicating with one another. So if these cells are not in direct contact, they will die. And this is an example from your heart cells, your cardiac cells. So if your heart cells are dying, you are dying. Now let's talk about autocrine and paracrine. So autocrine, simply the cell signals to itself. It sends a ligand and binds to itself. The paracrine, it's very similar. The only difference is that the receptor is on a local cell, so a cell within its vicinity. And let's look at this example. So we have tumor cells, which come from cancer, and they release this ligand called VEGF. So VEGF will bind to this receptor called FIT4. It can do it on a tumor cell, and that would be autocrine, or it can do so on the lymphatic endothelial cell, and that would be paracrine, because they're within the vicinity. You don't need to know specific examples, so please do not emphasize on them. All you need to, as long as you understand that this would be autocrine and this would be paracrine, that's good enough for me. And that's good enough for your exam. What about the long distance ones, the neuronal and endocrine? So neuronal, we said, involves neurons. So you have an action potential, it travels along the axon, releases neurotransmitters, and that hits your target cell. And then we have endocrine, which is basically the hormones released to the bloodstream and reach the target cell. Now, they are both long distance. However, neuronal is special in the sense that it is both long and short distance. How? Nerves are connected to each other, right? So a nerve communicating with another nerve is neuronal communication. But a nerve all the way from like your spine, all the way to your finger, that is quite a long distance. So that would be an example of long distance. And here's a summary. So we have juxtacrine, which is direct contact, autocrine, ligand to its own receptor, paracrine to a local receptor, neurocrine to neurons, and endocrine, hormones to the bloodstream. Now, I said there was a basketball example of this. So let's think of juxtacrine as kind of like when you have the ball and you just hand it off to your teammate. Autocrine is when you throw the ball to yourself, and that would be a travel. Paracrine is kind of just a short toss or a short pass to your teammate. Endocrine, I'll get to neurocrine by the way later, but endocrine is basically when you throw the ball a long distance all the way from half court to your teammate at the end zone. And then you have neurocrine. This is basically kind of like, it can either be this huge long distance, but it takes a series of passes. That may have confused you, so just as long as you understand this, that's good enough. All right, any questions? And Hamza, can you please check chat for any questions?
No questions.
All right, so everyone's clear on this. All right, yeah, let's move on then. Again, if I'm going too fast, or if I'm going too slow, or if something's not making sense, please say so. I tend to accidentally go a little too fast sometimes, so I apologize in advance if that happens. Let's move on to a question, because that's the way we test our knowledge. Contact-dependent communication between two cells using gap junctions. Which of the following describes this method of signaling? All right, so you can answer this in the chat. All right, so some people are saying A, seeing a lot of A. Does anyone think it's B, C, D, E, F maybe? All right, so everyone's saying it's A, and that's correct. This is juxtacrine communication. contact dependent. So as I said, contact dependent because they're directly touching each other. And if they're not, they'll die. Okay, let's move on to another short objective, the principles of cell signaling. So of course, because people like to philosophize and falsaf, we have to have these principles, but they are very important because this will be crucial to our understanding of the cyclic AMP and IP3 pathways. So how does cell signaling work? First of all, the signal is not going to be in one form. We need to convert signals from one form to another, and that process is called transduction. So if any of you play like electric guitar, for example, when you'll basically like strum the strings, but it's not the mechanical, you have to convert it into electrical or sound energy. So you're basically strumming, and then the strings will transduce that energy or that signal into like sound or electricity if you're using electric, right? Now, why is this important? Well, for one, a molecule by itself not do anything unless it's transduced. But another thing is that the same ligand, can interact with different cells. So why is this important? Sometimes we have one ligand that's capable of different functions, but it's the way it's transduced which allows those different functions. So transduction allows ligands to interact with different cells and therefore produce different functions. Let's look at this example of heart muscle cells. So acetylcholine, when it binds to a heart muscle cell, it will decrease the rate and force of contraction. But when it binds to your salivary gland cells, it will cause secretion. Let me ask you something. Do your heart muscle cells secrete anything, or do they secrete saliva? No, they don't. That would be very concerning. Okay, what is a ligand? A ligand is just a signal molecule. All right. So yeah, if our heart muscle cells were secreting saliva, there'd be a problem. Our heart muscle cells, when acetylcholine binds to them, they need to do something else. And that would be decreasing the rate and force of contraction. Now you're probably wondering, how does this happen? That is way too advanced for now. And we really do not want to get into those details. Let's move on to the next principle, which is intracellular and surface binding. So what kinds of substances can go through the membrane without any channels? Please answer in the chat. Non-polar, all right, what? All right, steroid, hydrophilic, gases, okay. So we're seeing a lot of different answers, but I finally found the one I like, which is either lipid soluble or hydrophobic. So hydrophobic, water repelling, will pass through the membrane without any channels. So if it passes through the membrane without any channels, what is the opposite substance and let me rephrase that. What kinds of substances need to go through channels? Polar, yeah. Polar, hydrophilic, exactly. So hydrophilic substances need channels. So yeah, now let's see how that works. The reason this is important is because most likely intracellular signals will be the ones that control transcription, all right? So let's say we have cortisol. Cortisol is a steroid and steroids are always hydrophobic. Cortisol is going to control transcription by going through the membrane, binding to a protein inside, and then controlling transcription through whichever way it does. And you guys have already seen the lecture and Mo about transcription. So I'm assuming you all know how it works. If not, we'll just keep it as process of making proteins, just to keep it simple. All right. Now, someone asked a question: how will hydrophobic pass through the hydrophilic head? Even though we have hydrophilic heads. Okay, let's think about it this way. Let's say we had a hydrophilic substance, all right? And a hydrophilic substance, it will get through the hydrophilic part, it will get through the hydrophilic heads, but the tails, the hydrophobic part, will repel it. And the hydrophobic parts are mainly what dominate whether or not something hydrophilic or phobic will pass through. So hydrophilic are more likely to be repelled in general, but that's all for like membrane physics, and really it's way too much for now. But I believe there was a lecture on this. If not, I can re-explain it. But what about what's it called? What about? No, that's about it. So let's move on to the just a recap. So again, we said transduction is important because it converts signals from one form to another, and it allows the same ligand to interact with different cells. Now, some ligands will bind on the surface; those are hydrophilic, and some will bind intracellularly. Those are hydrophobic, and those intracellular ones will control transcription. All right, does that sound clear to everyone? All right, great. We'll move on to any questions. Again, anything that didn't make sense. So Hamza, can you check chat for me, please?
There's no questions so far.
All right, then I have a question for you guys. Which pairs are correct? A, B, C, or D? So you can answer in chat if you'd like. All right, so yes, B is correct. And is there another correct answer? C, very good. B and C are both correct. All right, so we said hydrophilic will bind on the surface and hydrophobic will bind intracellularly. All right, does anyone not understand why that happens? Great. Okay, see how? Okay, so let's go through it. So let's go back to our old diagram right here. Again, we said that the plasma membrane will only allow substances which are hydrophobic to pass through without any channels. This means hydrophilic needs surface binding and hydrophobic will bind in the intracellular, all right? Because it can pass through the plasma membrane passively without any sort of channel. All right, I think we can move on to the next objective, which is molecular switches and speed. Now, the whole speed part is not that big of a deal. All you need to know is that the fast signals will alter function and the slow ones will alter expression. Why is this important? That's not really discussed that much. The slide was kind of just there and I felt the need to include it just in case. But what is more important and what is crucial. So if you weren't paying attention with me before, I'd ask that you start paying attention now. And this is the molecular switches. We have two types, kinases and GTP binding proteins. So we have these proteins called kinases. Kinases are these proteins that are these enzymes that transfer energy. And they do so by hydrolyzing ATP. What's going to happen in a kinase switch? So first we have a protein that is inactivated, and then you get the signal to turn it on. The kinase will be activated, and when the kinase is activated, it will break down ATP into ADP and phosphate. So ATP will be broken down to ADP and phosphate. The phosphate's going to bind to this inactive protein and then activate it. So let's just recap. The protein right here is off. You get the signal to turn it on, and this will activate the kinase. The kinase will hydrolyze ATP to ADP and phosphates, and the phosphate will bind to this inactive protein to activate. How do we switch the signal off? We use a phosphatase, which basically just removes the phosphates. Does that seem clear? Because we are going to repeat it again. I have another slide where we're going to repeat this again. All right, so it seems clear, but just so everyone's on the same page, we have it in words because everybody loves reading. So kinase, again, this means it transfers high energy. It hydrolyzes ATP. So first the protein receives a signal to turn on, and then the kinase will hydrolyze the ATP into ADP and phosphates. The phosphate will bind to the protein and this activates it. And then when we want to turn it off, we use a phosphatase. Now again, focus more on the activation because this is where the questions are more likely to come from. The inactivation is very easy. So it's honestly a very easy detail. You're just removing it or you're breaking it in both steps. Right. What about GTP binding proteins? First, we have this protein called a GTP binding protein. So as the name implies, it's going to bind GTP. So in the inactive state of this protein, it's bound to GDP. So we get this signal to turn the protein on and GDP will be released. In its place, we have GTP, guanosine triphosphates, which will bind to this protein to activate it. And then when we want to turn the signal off, we break GTP and into GDP and phosphate. All right, so let's just recap. The G binding protein is off because it has GDP bound to it. When it gets the signal to turn on, GDP is released and GTP comes in its place. When GTP comes, it will activate the protein and it sends a signal out. When we want to turn this off, we will hydrolyze the GTP. Does that make sense? All right, we're gonna go through it one more time just in case. Okay, what do you, okay, what about ATP? What do you mean? Could you please clarify? All right, I'll wait on the clarification, but in the meantime, let's go through the words of this. So GTP binding protein means it needs to bind GTP to activate it. So the protein will receive a signal to turn it on. The GDP, which is originally bound, will be released and replaced by GTP. When GTP binds to the protein will be activated. And when we want to turn off this protein, we hydrolyze GTP. We hydrolyze GTP into GDP and phosphate. Now, if you want a funny way to remember this, this was an example given to me by a student in pre-med about good friends and bad friends. So right now you are sad. You are this protein right here. You're a sad little protein and you have a bad friend, a toxic friend, all right? That's GDP. One day your mom starts yelling at you, telling you to change your life. You need to be a better person. So that's a signal to activate and you get rid of this bad friend and you get a good friend instead, GTP. So GTP makes you active, makes you happy. You start signaling out that like, look at me, I'm a better person. And when we want to turn the signal off, when you seem too happy, turns out your toxic friend had a bad effect on you and you just break them down into GDP and you're inactive again. So is that all clear? All right, great. Okay, where does phosphate once it's hydrolyzed? Okay, so, okay, I don't know why it does that, but okay, the phosphate will not be hydrolyzed for the record. The phosphate, when it leaves, it kind of just exists within the interstitium. It might bind to, it might go back with ADP to make ATP. Honestly, God knows what it will do, to be frank with you. But it could be involved in many different things. So is that clear? Great. Okay. I do not have a question. Now, I want to ask you guys, do you want a 5-minute break? Because this is where things start to get difficult. So we can have a 5-minute break to answer questions, or we can just move on. Okay, so none of you
asked, yeah. What's the difference other than ATP and GDP?
Wait, could you repeat that, please?
What's the difference? What's exactly the difference other than ATP and GDP? I don't.
You mean between the switches?
Could you clarify the question?
Wait, could you just could you just clarify what you mean? Because sorry, I'm a little confused. All right. So I'm just gonna keep the slide open, but okay, what's the difference? Oh, okay, so they're not going to ask you a question, what's the difference between this switch and this switch directly. These switches, again, the reason I'm explaining this is because it's going to build you a basis for what comes next, all right? But in case you want to know the difference, so kinases, again, the switch is called a kinase switch. The kinase switch will hydrolyze ATP and bind phosphate to activate the protein. The GTP binding protein will release GDP and bind GTP to activate the protein. That's to keep it simple. But they're not going to ask you like the difference between ATP and GTP. It's just the molecule itself. And plus, it's not really something covered. All right, can you repeat the signaling? Okay, re-explain kinases. Of course, I can do that. So let's go through them again. Let's go through kinases. Again, as I said, a kinase is an enzyme that will hydrolyze ATP or transfer energy, whichever way you want to think about it. How will this work? You have this protein right here and it's inactivated. So when you want to activate it, you use this kinase to break the ATP into ADP and another molecule called a phosphate. You can also think of it as P or PI. So when this phosphate is released, what happens is that it will bind to this inactive protein and therefore activates. So this protein will be activated and will start signaling out. When we want to turn off this protein, we release the phosphate by a phosphatase. That's kinase signaling. Does that make sense? All right. And yeah, another way to think about this is phosphorylation, but I feel like phosphorylation is just such an annoying word to use. So I just stick with the kinase switch. All right, now, are we all on the same page? Because we really need to be on the same page for this next objective. So if there's anything you're lost about, anything that's confusing, just say so now, let's get out of the way, and then we can move on. All good. All right. So I'm just going to assume that these two people speak for all of you, and let's move on. So today's main topic at least, is G protein linked receptors or G protein coupled receptors. Ion channels, enzyme linked, we don't care about that today. We care about G protein linked receptors. Now, before I get into that, I want to talk about something called a pathway. Now, a lot of you may not have taken what a pathway is before, and a lot of you may have. So just so everyone's on the same page, a pathway is defined as a series of consecutive reactions that a molecule takes to produce a final product. In simple terms, A and B will make C, with D will make E, etc. That's to keep it simple. Now, the funny thing about these pathways is that these individual steps can be involved in different processes. So we might be making pencils, but there's a step in making pencils that's responsible for making pens, or a step responsible for making erasers. Now, what's the nice thing about pathways and what's important to you as physiologists in the future is the fact that we have more steps. All right, so the more steps we have more places that need regulation. And the more places we need regulation, the more places there are for error. This right here is going to be very crucial because when we get into the cycle of AMP and IP3 pathways, I'm going to show you where things can go wrong and what can happen. And if you understand how that works, physiology becomes a lot easier. Let's talk about G protein coupled receptors. Now you might see these details and think that they're quite low yield. To that, I agree with you. However, they do tend to be asked about, especially this detail right here. So what are they? This is the largest class of cell surface receptors. So this is a receptor. And we said a receptor will bind a ligand. So this is the largest class of cell surface receptors, and it's ubiquitous towards ligands. What is this? This is a funny word that I use just because it was used in Dr. Shawaf's lecture. And because at some point there was a word limit, but ubiquitous basically means that it can accept many sorts of different ligands. It can accept amino acids, nucleotides, maybe even your mother. Well, maybe not that far, but my point is the ligands that this molecule can accept are quite diverse. Now, the characteristic feature of G protein coupled receptors is the fact that they cross the membrane 7 times. So let's count together. 1, 2, 3, 4, 5, 6, 7. Wow, okay, great. So another feature is that they are coupled with a G protein. So we have this G protein right here. This specific one has 3 subunits, alpha, beta, and gamma. Now the alpha unit is bound to GDP. Okay, GDP, we're going back to GDP. Where did we mention GDP earlier in this lecture? Okay, and just so we're all clear, ubiquitous basically means it isn't very specific. It's not very specific. It can accept many different ligands. All right, yeah. So we mentioned GDP in GTP binding proteins, all right? So we said GTP binding protein, we're going to see this GTP binding protein switch, all right? So again, this is one of the main reasons why I needed you guys' attention in molecular switches. Okay, let's look at how these G protein coupled receptors work. So we see this pathway, wow. And basically we have these hormones which bind to the GPCR, and this will trigger a change, all right? What does this change involve? This will involve activating the GTP binding protein. So you guys have to tell me, how do we activate a GTP binding protein? Simply. Via messenger, yes. Yeah, we remove GDP and add GTP. All right. So very good. So yeah, basically we're activating this. So this receptor or this molecule is going to be activated. This molecule right here is going to be activated by taking off this GDP and putting GTP in its place. All right. So we said that GDP was originally bound to the alpha subunit. So therefore GTP will bind to this alpha subunit. All right. And when GTP binds to the alpha subunit, so GTP will be here instead, the alpha subunit and GTP will be sequestered. This means they are shot to the target protein, all right? And then we said that there's a signal cascade. So this means that whatever signaling it's meant to do will happen. And then we terminate that signal through hydrolyzing GTP. But this isn't too important, all right? So let's just repeat. The hormone binds to the GPCR, or the ligand binds to the GPCR. This triggers a change. This activates the GTP binding protein. How? By releasing GDP and binding GTP. So GTP will bind to the alpha subunits, and the alpha subunit and GTP will be sequestered to the target protein. All right. This will cause a signal cascade, and then we can terminate the signal through GTP hydrolysis. Now you might be thinking, why do I have some words in bold? Well, that's because when I'm memorizing pathways, I like to focus on the main verb because that will help me remember it more. All right. So is everyone, yeah, and GPCR is the thing that, yes, it is right here. GPCRs are the ones that have these 7 transmembrane receptors, right? Now this was actually a detail that was asked about previously. So I really would suggest remembering at least that 7 part. Okay, what is sequestered? The alpha subunits and GTP. So basically this kind of breaks off. So the alpha subunit will kind of break off from this molecule along with GTP and be sequestered to the target protein. All right, is there anything else that doesn't make sense? Okay, just so we're all clear, we're going to repeat one more time because this is very important. And this time we're going to use my very low quality animation skills and the reason I don't have a job at Pixar. You'll see it very soon. So again, we said we have this ligand. This is going to bind to the GPCR. That's going to trigger a change. This change is going to involve bringing the G protein close to it, okay? This detail isn't that important. What's important is that we have an inactive G protein, okay? How do we activate this G protein? Someone tell me in the chat, how do we activate G proteins again? GTP. Yeah. Okay, so we're going to release this GDP. It's going to go away. And in its place, we're going to get GTP, okay? Now here you're going to see the terrible animation. What happens is that the GTP and the alpha subunit will be sequestered. Now, my goal was to try and make these go together. However, it completely failed and I haven't been able to fix it. When the GTP and the alpha subunit go to the target protein, it's going to cause a signal cascade. And that's basically, and if we want to turn this off, we release GTP, we break GTP into GDP and phosphates, and that's basically G protein coupled receptors. So is everyone clear on this? The target protein's in the cell membrane. Okay, so the target protein, yes, it's embedded within the cell membrane. But this is the inside of the cell. This is the outside of the cell. So all this is happening intracellularly. What does signal cascade mean? What does signal cascade mean? This basically means that you're just going to cause a signal or whatever response you're triggering is a cascade. So you'll have one thing that leads to the next, that leads to the next, that leads to the next. And you'll see that in the examples of cyclic AMP and IP3. Okay, the target is adenylyl cyclase. Okay, the target can be adenylyl cyclase, but in this example, it's just a general target protein. We have 2 target proteins we'll talk about in the examples. What does sequestered mean? Sequestered is, okay, so let me use my terrible animation skills again. Sequestered is basically this. When the alpha and the GTP are sent to the target protein, but they use the word sequestered because there's no other right word for some reason. What happens to beta and gamma? Beta and gamma kind of just exist. They kind of just slide there. Nothing really like significant happens to them, if I'm being honest with you. Right? What does sequestered mean? What does sequestered mean? Okay, again, sequestered, think about it as just the word sent, okay? So sequester, you send the alpha and the GTP to the target protein. All right. Is there anything else that's confusing you people? All right, nothing else. Again, we can repeat this again because it is very important. Because trust me, if you understand this, this is going to save us a lot of time. Easy peasy. I like that mentality. I love that mentality right there. Y'all hope the exams like that for you guys. Okay, to off, we make GDP into GTP. Okay, we don't make GDP GTP. We just take out GDP and put GTP in its place. And this would activate the G protein. Okay, are we all clear? Does GTP not get consumed or used up? Okay, so again, the whole idea of like GTP being used up, this is more something you'll take later on in MOL. I will repeat again right now, but basically the way that you use these in this case, we're not using GTP as an energy source, we're using it as an activating source. When we're using it for energy, we hydrolyze it because the bonds have phosphate, but don't focus on these details because they will annoy you. And again, it's not something that's exam based, but yeah, it's a little tricky concept for this level at least. So let's just repeat again. I'm not going to use my animations. I'm just going to stick with the words because I think it's a lot easier for everyone, including myself. So just to repeat one more time, a ligand will bind to the GPCR. This triggers a change. The G binding protein will be activated, and we activate it by binding GTP after releasing GDP. Once we bind GTP, it will bind to the alpha subunits, all right? And when it binds to the alpha subunit and GTP will be sequestered or sent to the target protein. This will cause a signal cascade, and a signal cascade is just a series of reactions that happen one another to get to a result. And then we terminate this signal by GTP hydrolysis into GDP and phosphates. All right, animation, we'll do whatever the people want, we will do. So let's go with animation one more time, just so we're all clear. The ligand right here will bind to the receptor. This causes a change, okay? This alpha subunit has GDP, we're going to release it. To activate this protein, we need GTP. When GTP binds to this protein, it's going to be sent, the alpha and the GTP will be sent to the target protein. This causes a signal cascade with that very fun 2007 animation. And then we can terminate the signal by hydrolyzing GTP into GDP and phosphate. So is everybody clear? Pixar's loss. I like to think that way. I appreciate the theses. All right, great, all clear. You're sure you all understand? Yes, alhamdulillah. All right, great, amazing. Right, now we're going again. No questions. Do you have any questions whatsoever? Because we need to really be on the same page. The next thing is going to be what makes everyone hate this lecture, including myself. Awesome. I'm going to assume Hiba speaks for all of you and just move on. And then, okay, so let's talk about cyclic AMP. Nothing on the slide is important besides what I have in bold. You may use cyclic AMP to control glycogen metabolism or gene expression. Glycogen metabolism or gene expression. Why is this important? Because I'm just going to give myself a mini drum roll. This right here. I believe this is what makes people hate the lecture. At least this is one of the things that made myself hate this lecture. So that's okay. We're going to make it simple, and I have everything in words. So we're going to repeat until it sticks with your head, because that's how you get through these things. You just have to keep repeating. So let's start. Everything up till here, we've already taken. So a ligand binds to the GPCR. This triggers a change. This will cause the GTP to bind to the alpha subunit. It's sequestered to the target protein, and it activates the signal cascade. Now, here, as one of your colleagues said, I believe it was Rima, that adenyl cyclase will be activated. This is the target protein, so we activated it. When we activate the target protein, this is what's going to cause this cascade. Right here, we have one thing leading to the next, leading to the next, leading to the next. So adenyl cyclase or adenylate cyclase will convert ATP into cyclic AMP. Just to keep it simple, adenyl cyclase will make cyclic AMP. Cyclic AMP will bind to this protein called protein kinase A. Cyclic AMP binds to protein kinase A to activate it. Again, there's a funny word there that we've taken before. Can anyone tell me what it is? Protein kinase A. There's one word here, kinase. Very good, math book. So kinase, all right, this protein is a kinase. So protein kinase A. That means it's going to hydrolyze ATP to ADP and use the phosphate to activate a protein, all right? So protein kinase A is going to activate phosphorylase kinase or protein phosphorylase kinase. Again, we have that funny word again. What's the funny word we have in protein phosphorylase kinase? Or what do we have in phosphorylase kinase? That's an interesting word that we care about. Kinase, very good. Again, we have kinase again. So this is another kinase. That means it's going to hydrolyze ATP into ADP, and it's going to use that phosphate to activate a protein. So the kinase will activate glycogen phosphorylase. Glycogen phosphorylase is going to do glycogen breakdown. So let's just repeat. So the GPCR will be activated after the ligand binds. This stuff we already taken. So I'm just going to go through it very quickly. The GPCR is activated. This is going to cause GDP to be released and GTP to bind to the alpha subunit of the G protein. The alpha subunit and GTP are going to be released or sent to adenyl cyclase. Adenyl cyclase will make cyclic AMP. Cyclic AMP will activate protein kinase A. Protein kinase A will activate phosphorylase kinase, and phosphorylase kinase will activate glycogen phosphorylase in order to do glycogen breakdown. Let's just keep it simple. So whenever there's a kinase, it hydrolyzes ATP. Exactly. Yes, that's the main point. That's the reason I wanted to emphasize so much on molecular switches, because they're everywhere. They're literally everywhere. We have a molecular switch, GTP binding protein, kinase. We have so many, and that's why it's so important. Should you memorize this? Aha. Now let me go through the other pathway and let me tell you how to study this. All right. But I do not believe you need to memorize it. I'll tell you what to do. So adrenaline, again, or ligand, these are the same exact steps as here. So we have the GPCR activated, the alpha subunit sequestered GTP to adenyl cyclase, makes cyclic AMP, activates protein kinase A. So we've already taken up till here. Now with protein kinase A, it's going to enter the nuclear membrane. All right, it's active, but it's going to enter the nuclear membrane. And we have this protein right here called transcription regulator. Because it's protein kinase, it's going to hydrolyze ATP into ADP and phosphate, and that phosphate is going to bind to the transcription regulator, which will allow this molecule to regulate transcription. All right. So just to repeat, cyclic AMP, just go through here. Cyclic AMP activates protein kinase A. Protein kinase A enters the nuclear membrane. This active protein kinase A will activate transcription regulator, and that will regulate transcription. All right. So, okay. After protein kinase A, sure. Let's go here. What happens after protein kinase A? And let's go here. What happens after protein kinase A? So protein kinase A is going to activate phosphorylase kinase. How does it activate it? Hydrolysis of ATP into ADP and phosphate binds to this inactive phosphorylase kinase. So once it activates it, phosphorylase kinase is going to do the same thing, but for a different protein. So phosphorylase kinase will activate glycogen phosphorylase. How? It's going to hydrolyze ATP into ADP and phosphate, and the phosphate will bind to glycogen phosphorylase. All right, that's for glycogen metabolism. For gene expression, cyclic AMP will bind to protein kinase A to activate it. The protein kinase A will enter the nuclear membrane, and once it does that, it's going to activate transcription regulator. How? Because it's a kinase, ATP into ADP and phosphates, and that phosphate will bind to transcription regulator to regulate transcription. All right. Can I go a bit slower? Sure. Okay. People's minds are fine. Okay, yeah, so we're going to have to go, okay. Some reason this isn't working. Okay, so let's go through this again, or let's try to keep this as simple as possible. Before I go through it again, I want to tell you guys how to actually study this. So instead of just going through the steps and plainly memorizing them, understand the way physiologists think. They're not going to ask you, what's supposed, or what normally happens here. They're not going to ask you stuff like that. They're going to ask you, where can things go wrong? All right. So when we look at the steps, as I said with pathways, we have different places where things can go wrong. Okay. So cyclic AMP, our second messenger, is going to be involved with glycogen breakdown and transcription. So most likely, if you get a question, let's say there's bad glycogen metabolism, what molecule is responsible? So it could be cyclic AMP, it could be protein kinase A, it could be phosphorylase kinase, or it can be glycogen phosphorylase. The steps are not as important to know as the specific molecules involved in that process. With transcription, it could be protein kinase A, it could be cyclic AMP, it could be transcription regulator. So understanding the molecules themselves, how they're involved, or sorry, not how they're involved, but understanding the molecules involved is more important than the steps. So we have, so let's just start from here. After adenylyl cyclase makes cyclic AMP, we want to do glycogen breakdown. So when cyclic AMP binds to protein kinase A, it's going to activate phosphorylase kinase. After phosphorylase kinase is active, it's going to activate another molecule, glycogen phosphorylase. And that's going to do glycogen breakdown. So cyclic AMP, protein kinase A, phosphorylase kinase, and glycogen phosphorylase are involved in glycogen breakdown. Right. What about transcription? We have cyclic AMP binding to protein kinase again. All right, protein kinase A will go all the way into the nuclear membrane, will activate transcription regulator. Simply, if it's going to control transcription, the molecule is called transcription regulator. When it activates that, it's going to do transcription regulation. So in transcription regulation, the molecules that are involved are cyclic AMP, protein kinase A, and transcription regulator. All right. Now, another thing that might help is knowing what specifically in this cyclic AMP pathway and what's specific in this pathway. So here we have two which are specific, phosphorylase kinase and glycogen phosphorylase. And then with here, we have protein kinase A, we have transcription regulator only. So the specific ones for glycogen breakdown are phosphorylase kinase and glycogen phosphorylase. For transcription regulation, it's transcription regulator. All right. How do you guys feel about this? All right, so, okay. Some people may still be having issues with this, and that's completely fine. This really, you have to give it time. Back when I was studying this, I gave cell communication a whole day. So I'd really recommend giving it the time it deserves. Gene expression. All right, so once mentioned the main ones, yes. What from it, what do we have to know? Second's complicated, all good right now. Gene expression is more difficult. What part they always ask about? The way they ask questions are mainly from the molecules themselves. They don't care as much about how it's activated, although it is important. They care more about what the molecules are involved in. So when we talk about cyclic AMP, right? Or let's say we're talking about glycogen breakdown, they're going to ask you a question. There's an issue with glycogen breakdown. What protein or what molecule is involved? Right here, you have 4 different molecules which could be involved. Cyclic AMP, protein kinase A, phosphorylase kinase, and glycogen phosphorylase. So we'll take out one of these, and that will be they'll give you one of these as an option, and then they'll also give you 3 other insignificant molecules. So they'll probably give you transcription regulator, they might give you phospholipase C, they might give you protein kinase C. So glycogen phosphorylase would be the right answer. And that's like how the questions are usually asked. If the signal is adrenaline for both, how does a cell know whether, okay, that's a very good question. So again, glycogen breakdown, this is not going, again, for one, let's ignore adrenaline just as an option, but it could be any ligand. The second way, there are these, again, it's very tiny processes that you really don't need to like pay that much attention to. And what's it called? Again, it could be different cells as well. So these two could be completely different cells. So again, it's all about how the signal itself is transduced and the specific changes that happen. But that's a very good question. Thank you for asking that. Okay. Okay, the second's complicated. I actually find the second one to be a little easier because you just have one specific molecule, which is transcription regulator. So as long as you understand that protein kinase, that cyclic AMP activates protein kinase A, which activates transcription regulator. And if there's an issue with transcription, which molecules could be involved, they'll take one of these and give you, for example, phosphorylase kinase or glycogen phosphorylase, and they'll ask you which one's involved with transcription regulation. That would be transcription regulator, to keep it simple. All right. Now, I know this is difficult, which is why I have everything in words, because words are important, all right? So if you're still having issues, go through these words, it'll make your life easier, okay? So the ligand will bind to the receptor, and that activates a G protein by sequestering the alpha subunit with GTP. When the target protein is activated, that's adenylyl cyclase or adenylate cyclase, the 4-ATP will be converted to cyclic AMP. Keep it simple, ligand binds to receptor, activates G protein, alpha subunit with GTP are sequestered, adenylyl cyclase is activated and makes cyclic AMP, right? So the whole goal of these GPCR steps are to make cyclic AMP, all right? Adenylyl cyclase will make cyclic AMP. But what about after glycogen metabolism? Now we have our two pathways. First, again, there's this kind note about how kinases work, right? So kinases will hydrolyze ATP to ADP and phosphates, and phosphate will bind to the inactive protein to activate it. Like, how does this work? Cyclic AMP will activate pKa. pKa activates protein phosphorylase kinase. Protein phosphorylase kinase activates glycogen phosphorylase, and glycogen phosphorylase does glycogen metabolism. Let's keep it simple. Cyclic AMP activates protein kinase A, which activates protein phosphorylase kinase, which activates glycogen phosphorylase, which does glycogen metabolism. How could questions come up? They will give you one of these proteins, or they will give you one of these 4 molecules. And they'll ask, and they can ask you a question about other molecules. They'll tell you there's an issue with glycogen metabolism. So they'll ask you which of these is malfunctioning. So is cyclic AMP malfunctioning? Is protein phosphorylase kinase malfunctioning? And they'll give you like 4 other insignificant proteins, and you just have to pick one of these, and you'll be fine. What about transcription? It's a lot easier. Again, that same kind note. Cyclic AMP activates protein kinase A, which enters the nuclear membrane to activate transcription regulator, and transcription is regulated. So let's say we have an issue with transcription. They will give you one of these proteins or one of these molecules and ask you which one, they'll ask you which one's malfunctioning, and they'll give you 4 others or 3 others. And then they'll tell you, okay, they'll give you one of these as an option, and they'll tell you which ones should be involved transcriptional control. That would be cyclic AMP, protein kinase A, transcription regulator. Like, does that make a little more sense than it used to? Okay, are you sure? All right, so ZK wants us to move on. If he wants to, then we will move on. All right, like IP3 and CA2+. This is our last example that you need to know. Well, one of 2 examples that you need to know, but yeah, again, very important. So what do we need to know from here? IP3, the goal of IP3 is to cause intracellular IC calcium release, all right? Now let's look at everyone's favorite pathway. Again, back when I was studying this part right here just confused me so much. So that's going to be the main part I go through so that you understand this. The presentation will be sent, the recording will be sent, you guys will have all the resources, so don't worry. First, we have the signal molecule. Again, everything up till here we've already taken. So ligand binds to GPCR, activates the G protein, sequesters alpha with GTP to phospholipase C, our target enzyme. Phospholipase C, PLC, will convert PIP2, which isn't shown here because it has a weird name, into diacylglycerol and inositol triphosphate, IP3 and DAG, IP3 and DAG by phospholipase C. Like, why do we have this weird fork-looking thing? So that's because this is representing PIP2 becoming diacylglycerol and IP3. Diacylglycerol, DAG, will bind to protein kinase C. It's involved in the activation, but it doesn't activate yet, and I'll get into that. Now, what about IP3? IP3 is going to bind to the calcium channels, and it will open these calcium channels, causing release of calcium, all right? And this release of calcium, along with diacylglycerol, will activate protein kinase C. So why is this important? Calcium has a lot of function, and it can only do so through protein kinase C. And protein kinase C has a lot of that function, right? Now, we don't have any fancy names to pay attention to in this pathway, which is probably better for you. However, let's just go through one more time. So again, we already know these steps, that the signal molecule will bind to the GPCR, triggers a change, alpha subunit with GTP sequestered, and then we get to phospholipase C, which breaks PIP2 into DAG and IP3. DAG, IP3. When that happens, the diacylglycerol will move along the membrane to protein kinase C, but it won't activate it yet. Why? That's because IP3, the other molecule which was released, has to bind to the calcium channels. This will cause them to open and a release of calcium. Together, DAG and calcium will activate protein kinase C. Right. There's something special you might notice. We actually have 3 intracellular messengers. Although it's called the IP3 pathway or the phospholipase C pathway, we actually have 3 messengers. We have IP3, diacylglycerol, and calcium. Calcium is an intracellular messenger as well, because an intracellular messenger is a messenger that responds to a signal before it. So we have a signal from the outside, and then we have these 2 molecules which were released from that outside signal. And calcium was released because of another signal. So we have 3 secondary messengers: diacylglycerol, IP3, and calcium. Like, let's repeat one more time. So, or actually, before we repeat, let's talk about how questions could be asked here. So we did actually have, I do remember a question being asked about this. So again, IP3 is responsible for calcium release. That's its function. Diacylglycerol and calcium are responsible for activating protein kinase C. Understanding this is very important. Also knowing the target enzyme, phospholipase C. Phospholipase C is a target enzyme in this case, and will break PIP2 into DAG and IP3. Like, how could questions come? Let's say they tell you something about intracellular calcium not being released, and they'll ask you what molecule or what step is most likely involved, right? And so they'll give you phospholipase C, they'll give you diacylglycerol, they'll give you protein kinase A, and they'll give you protein phosphorylase kinase. The answer would be phospholipase C, because phospholipase C makes IP3, which opens up calcium channels to cause their release. So if you understand that principle, everything becomes easier. Is everyone clear about that? Okay, what does phospholipase C break, and what is the goal of this pathway? Okay, the goal of this pathway is to activate protein kinase C. Protein kinase C can do many different things that we don't need to pay attention to now. You'll see some of it in MSK. But phospholipase C will break a molecule called PIP2. I'm going to type it in the chat, PIP2. Okay, it's not letting me type in chat for some reason. So PIP2, all right? So this molecule right here is PIP2. Phospholipase C breaks down PIP2 into DAG and IP3. Now let's just repeat the steps one more time. The GPCR is activated. This sequesters the alpha subunit with GTP. This is going to bind to phospholipase C. Phospholipase C is going to break it's going to break PIP2 into DAG and IP3. DAG is going to bind to protein kinase C, but it cannot activate it yet. We have to wait on another molecule. IP3 will bind to the calcium channels. This will cause the release of calcium, and that will bind to protein kinase C. Together, diacylglycerol and calcium will activate protein kinase C. Is that clear? Okay, can you repeat the question that came from the exam? Okay, does phospholipase C inhibit calcium release? No, it does not. Phospholipase C increases calcium release. All right, and the question that came on the exam, it was basically asking you that calcium is not being released intracellularly, They asked you which enzyme was most likely involved. So they gave us 4 enzymes, and one of them was phospholipase C. So phospholipase C was the right answer. What happens after PKC is active? Okay, is PIP2 inositol phospholipid? It is not. PIP2 has such a weird name that I'm going to be very honest with you. I tried saying it. I honestly just couldn't. So I didn't bother. Inositol phospholipid is mainly this IP3. But we mainly call it inositol triphosphate or just IP3. Like, what happens after PKC is activated? Does a cascade reaction take place? So most likely, yes, there will probably be a cascade reaction because of PKC. However, we don't need to worry about that for now. Okay, so we just know it as PIP2. Yes, you just need to know it as PIP2. They're not going to give you the full name. Most of the time, they'll keep it as IP3 or DAG or even PLC. They like to abbreviate a lot, so don't worry. Like, are there any other questions before we move on? Can you show PIP2 again? Okay, PIP2 is this right here. This molecule right here is PIP2, right? But for some reason, it didn't label it. Not quite sure why. But yeah, so I can't use my browser right now, but if someone could look, okay, the abbreviation really confuses you. Apologize for that. But fortunately, that's the way some things are. Are there any clinical questions related to this pathway? So not directly. The only clinical thing they may ask, so unless you've taken anything about calcium, which I don't think you have, they'll only mention calcium release. They won't mention like muscle contraction. Yeah, so it's phosphatidyl inositol 4,5-bisphosphate. It is way too long. Just stick with PIP2. But thank you, Haran. I'm assuming that's right. But yeah, clinical questions, again, they're going to explicitly mention this release of calcium because I don't think you've taken anything about like contraction or cardiac muscle cells. So yeah, is this only for MCQ? Yeah, these questions are more likely to appear on MCQ. Okay, what is the first messengers here? So first, again, the very first messenger is this ligand right here. There's no specific one that you need to know. It's just a ligand. But the secondary messengers, which are more important, are diacylglycerol, inositol triphosphate, and calcium, DAG, IP3, and Ca2+. All right, is everyone clear on that? All good? All right, all clear? Yes, yep, yes, yep. Very nice, very nice, very nice. In case we're not, fear not, I have words again. Everyone's favorite thing to do, look at words all day. Can you repeat the secondary messengers for glycogen metabolism, please? Is there something like this written part? Yes, there is. I'm on that right now. Okay, let me go back to the glycogen metabolism, and then we can come back to the what's it called? The written steps. So glycogen metabolism, the secondary messenger is cyclic AMP. This is the only secondary messenger. But the proteins involved are pKa, phosphorylase kinase, and glycogen phosphorylase. These are the 3 enzymes involved, or at least directly involved. So let's go back to the, what's the difference between primary and secondary pathways? Okay, so secondary pathways respond to primary pathways. That's what you really need to know. Or secondary messengers respond to primary messengers. So primary messengers are just like this first thing that's released, but don't worry about that. Worry more about secondary messengers, which are messengers that respond to these primary messengers. Like, let's go through words because everyone loves words. So the ligand will bind to the receptor, and this activates the G protein. How? Sequestering the alpha subunit with GTP. Like, the target protein, which is phospholipase C or PLC, is activated. And then PIP2 is converted into IP3 and DAG. Whatever that big word was, is converted into inositol triphosphates and diacylglycerol. All right. So again, we see these GPCR steps. Main thing to know, phospholipase C converts PIP2 into IP3 and diacylglycerol. Right. What about the calcium steps? So first, diacylglycerol is lipophilic, so it goes along the membrane to protein kinase C. Keep it simple, diacylglycerol goes to protein kinase C. All right. Then IP3 will bind to the calcium channels. The calcium channels will open, causing the release of calcium, and calcium will bind to protein kinase C, and together, DAG and calcium will activate it. So protein kinase C is activated by calcium and diacylglycerol. And just to repeat, diacylglycerol will go to protein kinase C, IP3 binds to the calcium channels, causing their release, and calcium and diacylglycerol will activate protein kinase C. Are we clear on that? What does transverse along the membrane mean? And what does DAG do? Okay, so transverse along the membrane. So let me just show you. Again, you can just think of it as goes to the membrane, but it's basically diacylglycerol is going to stay on the membrane, and then it gets protein kinase C. That's basically all this means. Don't worry about that. And as for protein kinase C, as for diacylglycerol, so diacylglycerol will basically, together with calcium, it will activate protein kinase C. So calcium, let me show you that right there. So calcium and diacylglycerol will activate protein kinase C. Right. Why is PIP2 not a secondary messenger? That's a very good question. So PIP2 is not a secondary messenger because it's not responding to a messenger. So PIP2 has to break to make secondary messengers, but it itself is not a messenger. All right, and OSPI, we can talk about that later. Right. Are there any other questions about PKC, IP3, anything in this lecture? What is PKC? Protein kinase C. What is DAG? DAG, diacylglycerol. So calcium and diacylglycerol will activate protein kinase C? Yes, you're absolutely right. All right, are there any other questions? Of course. Clear, thank you so much, thank you. No other questions, nice. Okay, well, of course, because I'm me and I have nothing better to do, I have some questions to ask you guys. So, cyclic AMP can exert many effects as a secondary messenger due to what enzyme? What is that enzyme? How can cyclic AMP exert its effects? Please answer in the chats. D-A. Okay, so I'm seeing a mix between A and D. Someone said B, A. Okay, so the correct answer is A, all right? Protein kinase A. Because, okay, let's look at the options. So cyclic AMP, first of all, protein kinase C and phospholipase C have nothing to do with cyclic AMP. So C and D are wrong. Protein phosphorylase kinase, although it is involved in this pathway, all right, it isn't responsible for every single thing. Protein phosphorylase kinase is responsible for glycogen metabolism. So it will act, so as we said, protein kinase A will activate protein phosphorylase kinase, and protein phosphorylase kinase will activate glycogen phosphorylase to do glycogen metabolism. Protein kinase A, though, can be involved with many different things. So we said it was involved with transcription, it was involved with glycogen metabolism, and there are many other examples, but it needs protein kinase A. So yeah, hope that makes sense. Now, is there anything that doesn't make sense in this lecture at all? Are you going to send the slide? Okay, so phospholipase C activates the calcium channels. Can you repeat, and are you going to send slides? Yes, the slides will be sent as soon as possible, so you will have them all. The recording has to take time to upload, but as soon as it's uploaded, you guys will have that. And can I repeat? Of course. So let me just repeat this. Cyclic AMP, first of all, protein kinase C and phospholipase C have nothing to do with cyclic AMP. So C and D are wrong. Cyclic AMP, the correct answer is protein kinase A, because B, protein phosphorylase kinase, is only involved with glycogen metabolism. Protein kinase A is involved with practically everything, all right? And phospholipase C activates the calcium channels. No, calcium channels will be activated by IP3. IP3 is made by phospholipase C breaking PIP2 into DAG and IP3. All right, is there anything else that's confusing you guys? Doesn't make sense? All good. Thank you. Thank you, guys. We still have, all right, we don't have any more lecture content to go through. However, I do have questions and I do have some summaries to go through, which will help you guys. So let's go through an exam style question with, of course, a little twist so we don't get copy striked by YouTube. So I'm going to read this question to you and you guys have to send me the correct answer in chat. So a medical student decides to spend Saudi National Day weekend on Mars instead of studying for their Foundation 111 final. Upon return, he spends 18 hours watching TikToks and then sleeps. He wakes up opening ExamSoft to see a cell communication question. After realizing that he had ignored the lecture, he passed out. The cardiologist said that his heart had stopped due to a lack of calcium being released intracellularly. Which enzyme is most likely malfunctioning? So send me the correct answer in chat, please. So some people are saying B, some people are saying D. Some people are saying B, some people are saying D, some people. Okay, some people are saying C, some people saying D. All right, we're seeing a lot of D, and yes, D is the correct answer. All right, let's go through how that all works. So, okay, first of all, we're talking about, again, there are a lot of unnecessary details here, like watching TikToks for 18 hours or waking up to a cell communication question. But what's more important is this sentence right here. His heart had stopped due to a lack of calcium being released intracellularly. This means we're looking at the calcium pathways. Immediately, we can eliminate A and B because protein kinase A has nothing to do with calcium. Protein phosphorylase kinase has nothing to do with calcium. Now we look at protein kinase C and phospholipase C. Now, protein kinase C responds to calcium. So if we said there was an issue, so we're looking at calcium as an option of protein kinase C not working, not the other way around. So the correct answer is D, because phospholipase C will make IP3, which increases intracellular calcium, right? That's how the question would work. So does that make sense to you guys? Yes. Yes, yep. All right, awesome. Or even MacBook is saying yes, so awesome. Yes, we need more questions like these. Repeat, please. Okay, so let me just repeat one more time. A lot of unnecessary details here. The most important thing is this sentence right here. So cardiologist said his heart stopped due to a lack of calcium being released intracellularly. The enzyme malfunction, and there's an enzyme malfunctioning. So calcium means A and B we're not even considering because they have nothing to do with calcium. D is the correct answer because phospholipase C will cause intracellular calcium release by making IP3, which binds to the calcium channels to cause their release. Protein kinase C responds to calcium. Calcium doesn't respond to protein kinase C. What's the difference between IP3 and D? I'm assuming, so yeah, so phospholipase C breaks down into GAG. Okay, phospholipase C will break PIP2 down into DAG and IP3. So IP3 will activate the calcium channels. If we had IP3 in the choices, do we choose it? Okay, so IP3 will not be a choice. And the reason is because we're asking for enzymes, all right? IP3 is just a secondary messenger. There's a difference between an enzyme and a secondary messenger. The enzyme is basically going to do the reaction and the messenger is going to signal. So let's say there was let's say I changed up the questions. Let's say I changed up the answers. Let's say I said, which molecule is most likely malfunctioning? And I put diacylglycerol, I put IP3, I put cyclic AMP, and I put amino acids, or no, I put carbohydrates. What would be the right answer? IP3, exactly. Because IP3 will cause the release of calcium. Very good, guys. Okay. So are there any other questions about this lecture? No, awesome. Okay, so now we have a summary, which I'm just going to go through quickly. And I just asked the person if they could please go on mute. All right, so cells can communicate through these methods: juxtacrine, autocrine, paracrine, endocrine, and neurocrine. Transduction allows ligands to diversify and function in specificity. G proteins are the largest class of receptors, and they have 7 transmembrane receptors. They function when the alpha subunit is sequestered to the target protein along with GTP. Cyclic AMP is involved with glycogen breakdown and gene transcription, and IP3 causes intracellular calcium release to activate protein kinase C. So these are some key points, but as usual, please go through the lecture. That's all I really have to say. Of course, I do have some suggested watches. I made a video about calcium and about IP3 diacylglycerol, about IP3 and cyclic AMP and GPCRs. So if you're still confused about that, here's a video where I draw everything out. Now, when I was studying this lecture, these were the videos I used to help me understand them. The resources I used were your PowerPoints, so big shout out to Dr. Jawaf. Of course, the osmosis cell communication is also an amazing video, highly suggested. And not a big fan of textbooks, but I wanted to make sure the information I was giving you was accurate. Now, of course, thank you.